Good afternoon and good morning, everyone. It's really a pleasure here to report. And the talk will be around 30 minutes. And feel free to interrupt me and we're in between for any questions. The talk will be around large models for single-cell omics and drug discovery. And I will start by a very quick summarization of scGPT and also a quick intro for single-cell foundation models for people who are not very familiar with this direction. And then followed by personal takes on several lessons learned after the release of scGPT. So first of all, what are single-cell omics and why we even care about building foundation model for it? Different from bulk-level readout, single-cell sequencing provides cellular scale resolution and reviews cell heterogeneities. For example, this can be conducted by droplet-based single-cell RNA-seq single-cell RNA-seq technology, where individual cells are barcoded into their RNA widths and then can be read and separated in later stage. Recent advancement enables multimodal sequencing across the entire central dogma. For example, you have ATAC-seq can be used to measure the chromatin accessibility, and SET-seq measures RNA expression and protein altogether. We also have perturb-seq, which is like CRISPR-based perturbation sequencing, that are combined the RNA sequencing with the perturbation, large-scale perturbation in different cells. So you can have all the information read in hash throughput. We can see that this reservoir of technologies now enable us to have this comprehensive view of individual cells and their states. So today we will more or less focus on the technology most related to RNA sequencing. And the reason for that is that it is, to date is most established technology. We have the data with single-cell RNA-seq in large abundance. You can find around a few hundred million of varying cells in public domains. And also they have to date a higher data quality when compared to other technologies. So these features really make it a suitable training data for a foundation model to answer relevant biological questions. Now, once we have a foundation model, or even nowadays we aim for virtual cell models. Now we can use it to answer many, if not all, questions in the field, from more biological related questions to more therapeutic related questions like predicting perturbational response or predicting the differentiation trajectories of cells. And more importantly, if we think about it, these applications, the different application tasks, they are interconnected. For example, as visualized here, solving one gene interactions may greatly contribute to the solution of other questions like how cell differentiates. So this justifies the rationale of building foundation models, as a model can learn from and provide a shared biological context for many different applications. So how did we build a single-cell foundation model? We first care about the data and model architecture choice. While texts are made up of words, cells can be characterized by genes. This inspires us to believe that transformers would be also suitable to learn interactions between the gene tokens. The self-attention can be also quite suitable here. And we also already have the community already have provided a vast scale of sequencing data, such as shown here on the right-hand side. This is a screenshot from the cell by gene census, which contains tens of million cells of sequenced single-cell data. So actually, this screenshot was taken from 2023, the first day when we began to train the scGPT model. The public domain really grows in accelerating speed over the last years, from 33 million cells in 2023 to around more than 100 million cells nowadays in this same cell by gene census collection. Now, therefore, one can train a large model via self-supervised learning to build a pre-trained model that understands cell types and cell states. We conducted an autoregressive generative pre-training. In the pre-training, large-scale expression matrices were collected and pre-processed. We selected around 33 million normal human cells of 51 organs from the collection, the cell by gene collection we just mentioned before. Our model differs from transformers in the natural language processing domain in terms of token embedding difference and also the transformer model itself. For the input embeddings, we use 3 sets of tokens to represent the single-cell RNA-seq data. First, we have the embeddings for gene identifiers, which maps all gene names to a vocabulary of learnable vectors. Second, we used a fully connected neural network to encode the expression value into embeddings. Since the expression value are continuous magnitude values. In the processing stage, we transform the expression value, using a technology we introduced called value binning, which is basically looking at a relative expression regarding each of the cells. And the third, we include a flexible condition token, denoted as this TC here. These are flags of possible metadata you want to introduce during the pre-training or fine-tuning in the later stage of model. For example, maybe you want to use that to introduce to identify whether this is a gene that is with a expression already known to the model, or this is a gene with unknown expression to be predicted or learned by this model. So the core model is a 12-stacked transformer layers, which with a customized mask attention strategy. So briefly speaking, this model is trained to reconstruct unseen gene expressions by this mask attention. And a mask attention guarantee that only right information can be seen for when we wanted to predict each of the gene. Only right information of other already known genes can be seen during the transformer layers. So the output of the model HL, represented here, is a group of vectors with a length equaling the number of input genes. We also reserve the special token at the beginning of the output, which is, we call it a CLS token. So the output at this position can represent the integrated information. In other words, it represents the cell embedding. scGPT can support varying applications after the pre-training. These tasks are achieved either by fine-tuning or in a zero-shot calling of the pre-trained model directly. I listed several different applications here. And so one of them I wanted to highlight it is a task we call reference mapping. We build this very fast reference nearest neighbor search using cell embeddings. So one can map and annotate cells by querying similar cells from a very large cell atlas. Since we can store the atlas embeddings, we just use a model, call on hundreds of million cells for once, and store all the computed cell embeddings. And also now we can encode new cells very efficiently using the pre-trained model, and then querying the cells with into all the stored cell embeddings. So this workflow makes the whole process very fast. One can use it to annotate cells to search for their nearest neighbors very fast on GPU within seconds. Here showing within seconds, 0.1 seconds for 4,000 queries. Another very important and interesting task we highlighted was the perturbational response prediction. So here the model was fine-tuned after the pre-training, and then it was optimized to learn to predict the unseen perturbations. So you learn from some combinations of perturbations and then try to predict the unseen ones. To predict the generative genetic perturbational response, the model was tested on 3 different benchmark datasets, trained on the training set and test on the test set of this 3 different datasets. We showed and visualized here that at that time our model performs the previous state-of-the-art method years and demonstrate the predictions here shown by 2 special cases of different genes that the model demonstrate predictions highly correlated with the ground truth post-perturbation response. So we also introduced a reverse perturbation task. So given the result cell state, we want a model to predict the source perturbations. So here are, this was showcased on a rather small subset of data of 7 test cases. Here on the left-hand side shows 2 test cases out of the 7, where the model successfully identified the predictor cases shows are the top 1 or top 2 cases, the correlators aligns with the actual perturbations. So the model learns from perturbation responses and try to guess, given a new perturbation response, what are the origin cause of the genes that are being knocked out for this result. On the overall, we see a rather higher hit rate is observed among all 7 test cases when compared to peers, and also compared to a baseline of calling the most differential expressed genes. To summarize, scGPT provides a general pre-training workflow for single-cell data and supports various tasks. We were quite fortunate that scGPT became a quite popular tool in the field, and it gave us a chance to communicate and learn lessons from our users. We learned several lessons. So first, scGPT indeed can generate biological meaningful cell and gene embeddings. Here I show an example of data of a very large retina atlas collected by a user group. So this was a relatively newer data set. We didn't have that during the pre-training. It was starting in the pre-training. You can see the pre-training embeddings. We're calling the pre-training embedding in a zero-shot way that those embeddings align very well with human annotations shown by the color, aligned well on all those 4 cell types. And our users saw over 95% accuracy of these labels, and are quite happy about it. This actually is a common case we saw also in other applications as well. But what they are not happy about is that if you look very carefully at more subtle cell types and subset of cells, this is what remains being missing here. So for example, here, this one I highlighted here is a very large blob of row cells. So row cells is actually a core cell type that can contain over 100 different annotated subtypes. So the pre-trained model does little job distinguishing them, apart. At least the model cannot distinguish them apart at this higher level. Now, for this very special cases, we actually collaborated very well with a user, so we train a fine-tuned model that can successfully see all the different small clusters of the subtypes. But I do want to point out that I recognize this missing piece, and that it surely can make more sense to have more ability of recognizing different cell states right after the pre-training. So that is still missing there. Another part is that we observed initial success about perturbational response prediction, as I shown in some early slides. This is also another image I borrowed from the GeneFormer paper. It also shows a successful pattern about instead of perturbation and reprogramming the FDS cells. However, in many other use cases we saw recently after the release of the model to the public, we found, generally speaking, the hit rate about finding a new unseen perturbation and accurately predict their response. This kind of hit rate can be low in real life. We still need more reliable predictions, especially for the pre-trained model of the shell. So one may suggest, we saw those kind of problems here. So one may suggest that maybe solving this problem, you can just go to the scaling up. If you scale up the model, we might be able to solve it. But what we also found is that it might not be a simple solution as we usually imagine. So here are some evidence about that. We originally reported the scaling in a published paper as well, particularly regarding here about the training data set. So if we enlarge the pre-training data size, we can see the performance of a particular application. So for here is a cell type annotation. We can see the performance of that actually growed along with the training data size of your pre-training. But actually, if you look carefully here, we saw a plateau after around 3 million cells. That means that after a certain stage, even if you increasing the data much larger, you didn't see a continuing large growing margin of your improvement. So this kind of phenomena, we gradually, and others I think also saw this kind of phenomena happen more frequently later for single cell foundation models. And this is actually not only for single cell. I also wanted to echo here, and for a phenomenon also reported for the protein language models. So this is some screenshot from Pro Gen 3, which is a more recent protein language model, where the researchers also found that when you having the different size of model, even you increase the parameter size of model a lot, but the performance on here is on the fitness score of the pre-trained model, it doesn't really improve after a certain stage. I think here is around that after 3 billion, the 10 billion or 46 billion models, they actually performed roughly the same. So we think, we examine this and also inspired by others, we think one of the key missing factor here is about task alignment. If you think about that in GPT-5 and GPT-4, in this natural language pre-trained models, the model was pre-trained to predict the next token. And then the model was used in a way to predict the next token. This is quite different for single cell foundation models and also for protein language models as well. I think this kind of a misalignment between the pre-training task and they are all the varying downstream tasks that are quite different from the pre-training task. This misalignment causes struggling on the scaling. There are potential solutions to that. I think one thing for us is about we need more line task. If you care about perturbation, having the perturbation prediction task right in your pre-training can maybe solve this problem. I will discuss more in later slides. So next is about with this reflections in my mind, we will discuss how the ongoing trend of multimodal and multitask retraining may address the alignment challenges and also see the recent trend of the field. So after the release of scGPT, G-Informer, and UCE, this early wave of single cell foundation models, we did saw a lot of new progress in the past year. They can be largely characterized in the 5 directions, I think, about multi-omics, dynamics modeling, about spatial omics, and also about integrating human knowledge into the pre-training, and about scaling up the model. We saw all a new wave of different methods coming up in the past year and in recent months. This also created a diverse view in the field, and people are using different strategies in the pre-training as well. Also, personally, collaborators and I also worked on some of these tasks, including MethylGPT, which is one of the work we used a scGPT style model, but on the epigenomics data of sequencing data. We also recently extended scGPT to the spatial domain, and we released a work called scGPT-spatial. We collected around 30 million data points and continually pre-trained the model for the spatial domain. And just in a summary view, we used the model again on different varying tests and showed quite good result comparing outperforming or incomparable with other state-of-the-art models. So although we have all this diversity of progress, it does make one wonder what is actually the most relevant next steps towards building a virtual cell models. So for me, the answer is about this direction, about dynamics modeling. For example, I mean dynamics modeling by you learn the cell differentiation, cell develop, also by learning the cell's behavior undergoing different perturbations. Now, why this is interesting, why I think, I personally think this can be the most important one. The reason are, first, the dynamics modeling task, they are quite deeply related to all the most important therapeutic questions and biological questions. So the same formula, imagine we're doing perturbational response prediction. We can have the original cell, we query the cell with the virtual cell model and query by, ask a model what kind of genes you want to perturb. You can use a model to virtually screen our different hundreds of thousands of genes and their combinations, and I use a model to encircle screening the results of the cells. And eventually you can find the most promising candidates of perturbations to give you a target cell state. This can be a super powerful tool to analyze, to understand the differentiation of cells And also the target cell state can be maybe, this can be a disease cell state, and this can be a recovers the disease cell to a more normal cell state. So that's a really powerful workflow. You can also apply that for therapeutics discoveries. Another angle to show the importance of this is that most of the, so scGPT was one example, and we also saw most of the even more recent methods, they are training the cell in one dimension of, we have different multimodal dimension of cell profiling, but they are in this one dimension of static profiling. You have a screenshot of the cell at a certain time point in this lifetime. But if you can train it to do the cell dynamics, it opens another new dimension. So they together, this orthogonal dimension can enable much more diverse applications. So to make it more possible, we also discuss the potential of enabling multimodal and dynamics training in a unified way by enabling different pre-training tests in a unified model, also unified text prompt. They can be put in a unified model architecture of hybrid transformers. In a recent perspective paper, we discussed this promising direction of training unified transformers and their hybrid multi-level attentions. You can imagine, so we imagine that the future of a much powerful model would be a model capable of looking at different layers of omics and also train the model in the way of, so this slide is showing that we can train the model in the way of unifying the different tasks. So the previous static task, like gene expression reconstruction, which is a task we use in pre-training scGPT, we can have that. We also can have other tasks like temporal predictions or perturbational training. They can be unified in the same way of prompting the model with different tokens. We are imagining some task tokens here. You input a token telling the model you're knocking off some genes, and another time you maybe prompt the model to telling, this time I'm training with a different data setting about temporal prediction to predict the next stage, the differentiation or development of the cell in a t+1 time. So in this, this is a perspective paper we envisioned of these possibilities about. It's actually quite straightforward and possible to use a unified framework with a ongoing diverse data to train the model to do much more things. That's one part. That's one direction about how we can enable perturbational training with the model, with a new modeling approaches. But after that, we found that the more interesting part can actually come to the data collection part. How we can be more efficient to use the active learning loop to make the data and the model co-evolve together in the same time. Because one of the bottlenecks currently, or I may say the most important bottleneck may be not about data, but about high quality and high volume data, about perturbational data, and about cell dynamics data. So so to illustrate this idea about how we can have model and data co-evolve together, let me introduce a recent work of us in the related but different field. We built a model. We built a model-based system for drug delivery, or to identify and discover new LNPs, lipid nanoparticles, for efficient mRNA transfection into cells. So we built a self-driving lab. This is a version 1 here, where you can actually see 3 different robots in the system. They containing all the consecutive steps from synthesizing the lipid nanoparticle, more importantly, synthesizing the different ionizable lipids in the lipid nanoparticle, and then formulating the particle and mRNA together, delivering them in cells, and also test them in vitro and read out and feedback to the model once again, all in this self-driving lab system. On the back end of the system, we also have a model. So this time, for this application, obviously, we have a chemical model, a chemical foundation model. And we use the data after pre-training of the model. We use the data collected in loops to fine-tune the model in an active learning way and to quickly find new targets. The system itself is also very high throughput. We achieve a throughput of 100 new RNPs per day per machine. Here is a 1-minute demo for that. We should be able to see the video now. So what you saw is that the system is actually in action, and it performs, all the plates are different combination of animals of lipids. It is actually doing the in vitro screening, but the candidates of the screening in every iteration are proposed by the model itself. So you have the model proposes candidates, and you synthesize the candidates. And more importantly, that you actually incubate the cell. You inject the candidates into the cell and then incubate the cell and read out in the same system in a closed loop. So with this system, so there's one more slide. We introduce the backend of the system. This is about the chemical foundation model. The interesting part is that it went through 3 different stages. Stage 1 is about a generic pre-training on generic small molecules, followed by a stage 2 of more lipid-like molecules, because the actual application is to find, using the model to propose ionizable lipids. And this stage 3 is just the video we just saw about actually applying the model in a active learning way of iterations of experiments. We did show the effectiveness of stage 1 of 2. The first column is a pre-trained model, and second 2 and 3 are the same architecture, but without some steps of the pre-training. We show the effectiveness of the pre-training and also the effectiveness of the whole model compared to other baselines of different architectures. So we use the model to work on exploring this space of a large chemical space. The space of chemical space is expanded by a library of different components. So each time you can pick up a promising candidates from one of the group and using them, putting them together in a one-spot chemistry to make a new lipid. So the model is, we optimize the model, learn to explore, which one of the lipids out of all this huge possible space of 220,000 models, and which one of them can be more promising to make a powerful new RNPs. And then, this is a visualization of that, of showing you pick up different components and then they can be made into a new lipid. To do that, we put the model in the iterations we just talked about. We do a active learning of 10 iterations. We also introduce a new strategy we call a balanced exploration and exploitation. So one branch of that is that you can have a plate of experiments, a plate of lipids you want to synthesize and test. And those are the plates of lipids that are with predictions that are most uncertain by your model. So it's a more traditionally active learning manner. But we also have another plate. We call it a exploitation plate. Those are the plates of the most promising candidates identified by by your model so far. So with this combination, we did saw that this strategy gives us a better efficiency for finding high-performing candidates in our simulations and experiments. For example, shown now here, this is the actual 10 experiments of results, and they are colored by the measured MTP, which is a transfection efficiency of the RNP, how effectively they can transfect mRNAs into cells. And you can see the steady increasing MTP, steady increasing result across different iterations. So eventually, very interestingly, is that the LUMI-lab, this is a name of the system, it not only identified very nice candidates, but also identifies this new strategy of bromination. This is the first time people actually introduce this bromine lipids into the lipids, but this is out of the top 6 lipids identified by the system is of this new family of brominated lipids. And in the first in vivo experiment, we did test the efficiency of the most promising RNPs in delivering candidates RNPs in mouse lung. And they showed very nice results by showing the best result when comparing to other strong baselines of MC3 and SM102. So this SM102 is an actual lipid nanoparticle using the COVID vaccine. And we also, in a head-to-head comparison, we saw that the yellow one, which is a LUMI-6, the most promising lipids with bromination, it shows the twofold increased efficiency when comparing to the LUMI-6D, which is almost the same lipids, but only missing the bromine. So showing this effectiveness strategy in a head-to-head comparison. If we go back to the story about training, about data and model co-evolve, and the LUMI-lab actually provide this empirical conclusion that it's actually quite possible to have this guided exploration and optimization of your model together to optimize, at least in the local minima, in a very local subspace of a very large space. So you don't really need hundreds of different iterations. If it's only in a very small specific application, it's very promising to do that in this co-evolve evolution. I just want to add a results from another recent paper as well, which is this evolve paper. It shows a similar system, but in a model-based directed evolution way. They applied that in some other areas, but also showed that similarly positive results of high efficiency. The point I want to make is that one can generalize this workflow to a single cell domain to perturbational training as well. We are currently in a stage of lacking really high quality and high volume data. We do have high volume data even in the public domain. Some people already released data about genome scale perturbation of certain cell types, usually some immortalized cell types, and with genome scale perturbation of 1 gene perturbations. So we can use that to train an individualization of a base model, like your version 0 model. This is like the base model we use in the LUMI-lab as well. And then you can use that to do more interesting things, to collect more interesting data. Say you are more interested, this is just an example, say you are interested in finding a co-stimulator to enhance your CAR T therapy. So for focused application, the researchers are usually more capable to give you a focused scope of the design. Maybe you are more interested not in genome-wide, but 1,000 different gene perturbations, candidate genes. And now you can do more interesting things. You can actually perturb the genes in, let's say, human T cells, not some immortalized T cell, but actual primary T cells. And you can also explore the combination of gene perturbations, not only single cell 1 gene perturbations. As long as you focus on more specific applications, you have this more freedom to generate high quality data, and high quality usually means more related with more in-depth task design and experiment design. So other thing can be visualized in these iterations of illustration. You have your hypothesis, you generate data, and you feed that into the model once again, and the model will give you a next round of hypothesis generation. This is exactly like what we did in LUMI-lab, and it's very possible to reproduce that in a single cell domain as well. I think I just want 1 or 2 more slides. So the great benefits or the potential great benefits is that you can do that in different applications. So for one application, you have maybe 10 or 20 iterations you accumulate some small scale, but quite important data that go beyond the existing cell line data. You might have more cherished data on primary cells or organoids. And in each one of the application, it's like you collected one island of data for that. And in general, all this outer loops of collection of different applications, we are collecting the most available data, the most available different diverse islands of data covering different aspects of the field. So this outer loop can be a more feasible approach in the short term that, when people are interested in a certain therapeutic application, it makes more sense to apply the current, version 1 or version 0 single cell model, virtual cell model, but applying the model together, not only in silico, but in this active learning loops with actual data collection and some data exploration. I mean that in the short term, I don't expect a model itself can solve out of the problem. But all these short-term mix-up elements are accumulating the data to train the version 2 and version 3, the ongoing new, more powerful virtual cell models. And in the long run, it might be actually laying the foundation to really give us the powerful models that can solve the most of problems in silico. Yeah. To summarize, what I showed before is that first about lessons learned from scGPT, and then the trends for building future virtual cells models, and particularly highlighting the trend about dynamics modeling. And one of the particularly interesting thing would be not only how we train the model, but how we can have the model and data co-evolve together in active learning groups. Thank you very much, everyone. And I'd like to express my deepest appreciation to all my collaborators and for their contributions.